How can two perfectly healthy parents have a child with a genetic disease? It's a deeply human mystery, right? Families have faced this literally throughout history. But the answer, well, it doesn't lie in anything we can see with the naked eye. It all comes down to a microscopic, high-stakes game of chance playing out deep inside ourselves.
Hey everyone, and welcome to This Explainer. I am absolutely thrilled to be your guide today as we journey into the incredibly fascinating mechanics of genetics. We're going to uncover the biological basis of inheritance, specifically looking at how our traits segregate and sort themselves out before a life even begins.
Okay, let's dive into this. Here is our roadmap. We'll start with the cellular secret of inheritance, move right into doing the genetic math, learn how to solve a test cross, explore real-world genetic counseling, and finally, we'll look at what happens when cell division actually fails. Section 1, the cellular secret of inheritance. To really understand why family traits seem to just disappear in one generation, only to mysteriously pop back up in the next, we have to look inside the cell at Mendel's law of segregation. This foundational rule dictates that each gamete, a single sperm or egg cell, receives exactly one allele of a gene pair. Now, this isn't magic. It happens because homologous chromosomes, those matching chromosome pairs you inherit from each parent, physically separate during a phase of sexual reproduction called meiosis 1. It's actually kind of mind-blowing to think Gregor Mendel figured out this massive rule of biology without ever actually seeing a chromosome. Now, what's really interesting here is the physical mechanism making this happen in anaphase 1. You can literally see the chiasmata resolving. That's the exact point where the chromosomes previously linked up. And those kinetochore microtubules, they're acting like tiny, incredibly powerful ropes. They attach to the full chromosome and physically pull the duplicated maternal and paternal alleles to completely opposite sides of the cell. They don't copy both into one cell, they literally split the pair. That physical separation is the absolute engine of inheritance. Section 2, doing the genetic math. So that physical pulling apart of chromosomes we just looked at, it directly translates into incredibly predictable mathematical rules. Think about it as a logical sequence. Step 1, because of that physical split, parental alleles separate with a clean 50-50 probability. It is essentially a microscopic coin toss. Step 2, those resulting gametes combine entirely at random during fertilization. And step 3, the resulting offspring's genotype, their underlying genetic code, determines the observable trait we actually end up seeing. Let's walk through this tree diagram to really visualize the math behind that sequence. If a parent carries a dominant big T allele and a recessive little t allele, the probability of passing on the big T is exactly 1/2. The probability of passing on the little t is also 1/2. So when two sex parents reproduce, you multiply those independent probabilities. 1/2 times 1/2 gives you a 1 in 4 or 25% chance for each specific fertilization event. Following the branches, you can see how it cleanly maps out into 4 distinct offspring possibilities. But, to really grasp inheritance, we have to emphasize the crucial difference between the invisible genetic code and the visible trait it produces. The genotypic ratio, the actual underlying alleles, is 1 big T to 2 big T to 1 little t. But the phenotypic ratio, what we actually see growing in the garden, is 3 tall plants to 1 short plant. Because the dominant T totally masks the recessive T, the invisible code looks quite a bit different from the visible reality. And this brilliantly illustrates how the Punnett square is just the perfect method for organizing those exact probability calculations. By placing the possible gametes from one parent along the top and the other along the side, we perfectly map out Mendel's famous 3-to-1 phenotypic ratio in heterozygous pea plants. It takes all that abstract math and shows us exactly why 3 of the offspring are tall and 1 is short. Section 3, solving the test cross. Now that we've got the math down, let's treat this like a detective story where we have to find a hidden gene. Imagine you're a breeder, okay? You have a tall plant. You know for sure it has at least one dominant big T allele. But is it purebred, homozygous? Or is it secretly carrying a recessive short gene? How do you actually determine an unknown dominant genotype? I invite you to pause for a second and predict how a breeder could actually figure this out. Well, the solution is a genetic test cross. You take your unknown plant and cross it with a homozygous recessive plant, a purely short little t plant. Compare outcome A and outcome B here. If your unknown parent was pure dominant, outcome A shows that 100% of the offspring will be tall. But if your unknown parent was hiding a recessive allele, outcome B reveals a 1-to-1 split, exactly half tall, half short offspring. Seeing that 50-50 split is absolute proof that the unknown parent was heterozygous. However, this is exactly where a lot of people make mistakes. Look at the student's initial incorrect prediction at the top. They assumed a test cross between a heterozygous parent and a totally recessive parent would yield that classic 1:2 to 1 F2 ratio. But looking at the true cross below, notice that the entirely recessive parent can only produce recessive gametes. Actually, scratch that. It's more like they just forgot to look at what the parent actually brings to the table. Therefore, no fully dominant offspring are mathematically possible. You absolutely must pay incredibly close attention to exactly what each parent can contribute. Let's firmly debunk a few more of these common misconceptions to make sure you don't fall into the same diploma trap that trips up so many biology students. First off, segregation is temporary for gamete formation. Alleles reunite at fertilization. Second, as we just saw, test crosses give 1:1 ratios, while F2 crosses between heterozygotes give 3:1 ratios. Third, Punnett squares show large-scale statistical probability, not a guaranteed outcome for a single family. And finally, that 3:1 ratio, it applies to all sexually reproducing organisms with complete dominance, not just Mendel's plants.
Section 4, Real-World Genetic Counseling. Now let's zoom out from pea plants to human application. 25%. This is a powerful number. It is the exact probability that two completely healthy, unaware carrier parents have of passing on a recessive genetic condition to their child. So the crucial point is when these principles apply to a disease like cystic fibrosis, two healthy carrier parents who have perfectly functional CFTR proteins themselves each carry one recessive allele. By random assortment, there is a 25% chance of an offspring inheriting both recessive alleles. At the molecular level, this means the child's cells only produce non-functional CFTR protein, resulting in the viscous mucus characteristic of the disease. Genetic counselors use this exact foundational math to help families understand their inheritance risks and make deeply informed decisions. Section 5. When cell division fails. We've seen how beautifully this system works when the rules are followed. But what happens when the cellular machinery actually breaks the law of segregation? We call this mechanical failure non-disjunction. Trace the error happening here during anaphase I. Instead of the homologous chromosomes being cleanly pulled apart to opposite poles, like we saw at the very beginning of our explainer, they both get dragged to the exact same pole. The cell has completely failed to segregate the chromosomes.
And the consequences of this microscopic error are incredibly severe. Because of non-disjunction, gametes end up receiving either 2 copies of a chromosome or 0 copies. When fertilization happens, this creates abnormal chromosome numbers, leaving a zygote with monosomy or trisomy, like trisomy 21, for example. This fundamentally disrupts the delicate gene dosage balance required for normal development, often causing profound developmental abnormalities, or even lethality. It really underlines the absolute delicate precision required for normal biological inheritance. So, what does this all mean? We started today with a mystery about how diseases pass from generation to generation, and we traced the answer all the way down to the physical tension of microtubules inside a dividing cell. It leaves us with a profound question. Do the microscopic odds of inheritance dictate our fate, or do they simply deal our starting hand? The biological basis of inheritance is a beautiful, fragile system of chance, mathematics, and molecular biology. I really hope you found this deep dive as fascinating as I do. Thanks so much for learning with me today.